facing pretty much straight into the current now, almost.
So wow, this whole area just looks like it's covered in manganese crust. It almost looks like it's forming mostly like a cement.
I'm lined up.
Yeah, copy. Is that coral or spread?
No, I think that's a really big sponge off in the distance.
Yep.
Or it might be a weird branching sponge.
Sponge.
Maybe it is.
So it's been interesting as we scan back and forth. We really aren't seeing all that much beyond sponges.
Nope. Seeing a couple more bamboo corals, but
Oh, look at that guy.
yeah, that's.
I know we've seen it
pretty
before,
awesome.
but like standing and like, that was the same one that we saw that had fallen over.
Yeah, so I think
same type.
maybe these. Chris, correct me if I'm wrong, but were you calling those stalked sponges that kind of flayed out the top? Were you calling them gramophone sponges on a previous expedition? Maybe I'm making that up.
230 or even further west.
I feel like that's something really crazy that I wouldn't make that up.
Yeah, I think 230's pretty good. Could go a little more west, but
Yeah, we got 5 more meters on this move. But
okay.
Working the thrusters today.
I mean, I think it could definitely be loose. It definitely looks like it could be some talus, like, you know, pieces that are not in situ that are covered in a manganese crust.
So I know Scott made this
Okay,
point
can I do
yesterday,
another,
but the geology
230?
and biology we've
Bridge
seen
nav?
today has been so different to what we saw on yesterday's
do
geode,
another,
which wasn't
just
far
like
from here,
130
as well as the
meters,
day
bearing
before's geode.
230,
And
0.2
all 3
knots.
geodes have been very, very different. And this just sort of shows the point that when you're
Thank
doing
you.
sort of baseline studies, one sample point is not enough. The more you have, the better you'll be able to characterize an area. If we were to have assumed that because all of these features are flat-topped, they would have the same biology, that we would be extremely wrong.
Yeah, there's some loose rocks here.
Thank you. And I'd also go with, we're also exploring just a very, very small part of each of these seamounts. So even though we're able to characterize one part of the seamount, I don't believe that we really can say that we know for certain what the rest of the seamount might look like.
Definitely, because you see zonation with all types of characteristics. So, for instance, because of depth, because of changes in temperature, current regime. And so we've seen that each of these seamounts in this one tiny area of each of those has been different from each other.
And that is, it is absolutely different than we thought it would be. I think that when we dove on the first seamount on Pigafetta, we naturally assumed that we would probably be seeing something that was much the same on the next two seamounts. So it's actually been quite interesting. Oh, look, another anemone.
Most certainly. So this looks like another Exocolactus, actinarian, or anemone.
So Scott and I had a fascinating conversation about these.
I missed that.
While you were out, I said, I believe that these are often identified by the morphology of their tentacles.
Right.
Because I've often heard you say that. But what Scott was saying was actually that you really need to collect a sample and look at the histology of the tissues to really make an identification about what kind of anemones these are. So I learned something new today, as I always do.
Can you guys hear me now?
Oh, only
We
a little
can
bit.
hear you.
You brighten up D2 a little bit? Yeah, thanks. Just trying to navigate.
Go for it, Scott.
I apologize for the delay. I can't hear you, so I was waiting to hear you on the video. I just wanted to follow up that very interesting conversation that you were having about not being able to characterize an entire seamount from a dive. And just to make this a little clearer to some of our viewers, we're only traveling about 8 football fields. I don't know how you would translate that into pitches for the international audience. Maybe you could do that, Diva.
No.
If you were to go to Mount Everest or somewhere in the Rockies and walk the distance of 8 football fields, you wouldn't assume you had seen everything that mountain had to offer.
That is an excellent point, Scott. And I think that really does bring it home in a way that we perhaps hadn't really made clear before. We're looking at really small areas of very large features on the seafloor. And it's certainly better than nothing, and it certainly supports the mission of this ship and of the NOAA Office of Ocean Exploration and Research, which is to characterize poorly known or unknown areas. But it can't get to what is the whole seamount based on less than 1 kilometer of track. But it helps.
Definitely.
So, Levi, if this gets any stronger,
Look at these dudes.
I'm not going to be able to move.
What do you think they're looking at?
The pillow lava.
Yay!
But
Pillows for
watch the
everybody.
thruster percentages.
Yeah, and if that's the case, I'm
This
gonna
has been our
turn to
most
port
abundant geological
and go
feature
into
so far.
But not
token
diverse.
configuration.
Okay, which is deeper water, so.
Well, yeah, it's deeper water, and it just means that you might have to come up a little bit, but
Okay.
we're good for right now, and it doesn't seem like it's going to get worse. So, but who knows?
Navigator, what's our current bailout plan?
Uh, 045.
Does that still sound good? Everybody?
I think it's.
I think it should be fine.
Yeah, it doesn't actually seem ideal, given that it's uphill in that direction. That's fine. That's the direction we've come from. That's,
Yeah.
yeah, that's good.
I think it's
Yeah,
the
that is
inverse
like-
of where
that is good.
right now. So
Yeah.
okay.
It's amazing how we keep going from these areas where it looks like all of a sudden, we have in place pillows, and we'll have,
we're
a lot
coming
of
into this move.
manganese crusts that sort of settle on top, and then
another
all
one,
of a sudden
Sam?
we'll turn around and we'll see an
Yeah,
area
there's
that looks
2
like it's
zero.
mostly tailless.
Yeah.
So, and it's interesting because they're not really dropping off the ledge
Bridge
left and
nav.
right. So, which is what
Let's
I
do
would
the
think
same
you'd be
move
more
again. 30
expecting to
meters
see.
during 230 0.2 knots.
Thanks for that.
Very interesting geology today. I think Diva didn't think I did a very good job explaining that one.
I always
Thank
think you
you.
do a great job, Deb.
Thank you, Diva. We aim to please.
Alright, let's come in video.
What are you aiming for? This?
Stock. Yeah, come in tight. I think we've seen this.
Oh, so this is a cladorized sponge, and it's another one of these predatory
Done here, pilot. I think
sponges.
there's a small coral.
Oh,
more or
maybe
less.
I'm wrong.
Cladorized
No,
sponge.
I am right. Okay.
And we actually saw a lot of these at the beginning
a
of our
lot
dive.
of these, yeah.
It took us a while to finally realize, it was actually when we zoomed in on another sponge that we realized that we were actually seeing a lot of these little tiny stalked sponges around.
When you're ready, video.
Okay, coming
What's
out.
the other guy you wanted? Where do you
Thanks.
see the coral?
Basically, keep tilting up. Okay, right next to the lasers now, just past it. Tilt down just a touch. There. And up.
I didn't
Come
even
in.
see that. Come on in.
Hey, Navigator, this is co-pilot.
Go ahead.
What time do we have to be off the bottom today?
About 1:30.
Okay, so
about
this is a
30 minutes.
chrysogorgid
Yep.
coral, probably from the genus Chrysogorgia. And it doesn't have any commensals in it.
Got one little red guy hanging on.
All right,
Oh,
come
really?
A little bit closer.
Yeah, and
Oh, yes.
Yeah.
Well spotted. What is that?
I don't know, but he's hanging on pretty tight.
Do we have any more zoom video?
No, that's all I got.
Huh. Doesn't look like a squat lobster.
I don't think so.
But yeah, it's holding on really tightly. Poor little guy.
Or maybe it's caught in the polyps?
Oh.
Like it's
Or-
being fed on.
Or a growth
in
in
a
the polyps?
polyp. Yeah, that's in a polyp. I can probably get closer. Here, come out partial.
Just so you're aware, pilot, I got you at 75 degrees.
Yeah.
You're good, but
Yeah.
just let you know.
Hey, come back in.
I got a little bit more.
Hey,
All right,
go ahead.
coming the rest of the way.
Not much clearer.
I think it's a little polychaete, a polynoid, so scale worm. I think I can see antennae. I mean, that doesn't really narrow it down. That could be anything with antennae, which could be a polychaete, an amphipod.
Come on out.
But I think it does look like a polychaete.
Full wide.
Thank you, pilots. Thank you, video.
Switch.
Looking
Okay.
straight down.
Hey, got a boogie. Another coral back there. Let's do a quick zoom video. I got a boogie.
Roger. Holding partial.
The sonar's got about 13 meters out, so
Quick zoom. Come in tight.
That's full. There it is.
And so this is another bamboo coral, or isidid. And this one's also got a chirostylid squat lobster.
Okay, I gotta move. I'm sorry.
It's okay.
Sitting on top of it, and you can see a couple of dead sponge stalks around there, covered in manganese coating,
Blackpool's hitting the ground, I think.
as well as a purple camatulid
pull up.
crinoid
Yeah, coming out.
that seem to be holding on for dear life. And as we look out into the distance, there are a couple of different types of sponges. Some polyopagons, some euplectellids.
Coming to the end of this move.
There we go.
Okay with another one, or do you want to?
Let me
catch-
push out
catch
a
our
little bit.
breath.
I got behind.
It's going to be a little while till Sirius actually stops, of course.
Ooh, and quite a big bolosoma sponge over there, bolosomine.
Okay, I'm comfortable now.
As well as we're passing a Tridopleura. Those are the leaf-like ones, just to the right. It's incredible. There's just hardly anything apart from these sponges.
That guy out there is huge.
Oh, and a couple crinoids. And yeah, another
That's a
really, really
sea
large
star.
polyopogon.
Maybe it's a crinoid. Come in on the sea star.
I was just about to ask what that was. So this is another one of the Henricea sea stars or asteroids. You can also see a couple more of those tiny sponges which we collected yesterday. Yesterday, I think. Yeah. They sort of appear to live in sort of a colony.
Right, come on in.
As well as another one of these really large polyopogon sponges.
If you have any more, go for it.
Right, that's full.
When you're happy, video.
Okay, a couple more seconds if we have it.
Lose my tail.
You're off to my starboard pilot.
Yeah.
Roger. Video's clear. Thank you very much.
The move is complete.
It's like every time you turn around, there's another sponge.
Just lurking. Lurking in the distance.
Whoa.
Ooh, Bolosomene.
Come in on the
So
head.
Chris was saying he thinks this is Bolosomene morphotype
Come
1
in on the head, video.
or unknown 1. And we collected Bolosomene morphotype 3, which is the ET sponge.
Stand by.
How interesting.
Yeah, so one of the reasons why we're not quite assuming it's a bolosoma in that genus Bolosoma is it doesn't have an osculum on the top, and the other two Bolosoma do, so it's a little bit curious. So for the moment, we're being a little bit conservative and just calling it in the stalked euplectellid subfamily Bolosomini.
Okay.
I'm looking back at you.
Yeah.
Thank you, Chris.
Just can't get ahead.
I feel like I've learned a lot about sponges in the last several days.
Me too. I'm going to have to say these are one of my favorite types of sponges.
They are very pretty.
Like sponge morphologies can just be so variable. It's amazing.
I don't know. I think I like the big, what are they called?
Polyopogons?
Yes, I like those. And we were talking, I think while you were out of the room, about the mesh that covers the oscula on those. We got some really beautiful close-ups on them.
Couple big heaves in there.
Just get some elevation for you.
Yep.
So if you're just joining us, we've got about a half an hour left in our dive. We're here at Delcano's Seamount, which is actually a geo, a flat-top seamount, where the flat top came from when this was once above the surface of the ocean. And it's been a really interesting dive because what we've seen on this, I think we both expected that we would have seen a lot of coral and sponges.
Yeah, definitely.
And we've seen very little coral. We've probably seen less than 10 individuals. And instead what we've seen is sponge after sponge after sponge. In fact, what we're looking at right now are dead sponges and live sponges all together. It's like a sponge party. And that's been a little bit surprising. But what's been very interesting about it, those were sponges and dead sponges. What's been really interesting has just simply been that we've seen a large diversity of sponges, but very few corals. And it's been very different from our other two seamount dives in the last 2 days. So,
thinking, especially if you get to that ridge.
lot to think about here.
Conservation of mass.
We are
right?
at a depth
Well,
of
I mean, that's not necessarily
1,822 meters right now.
Typically, that's what you see.
But I wouldn't be surprised if the higher we got, the higher the current got.
Is stable, but the
So now we're
antenna at the on the ship, the level of the carrier from the ship does appear to be moving, and that's
Another one of the
contrary
dead
to
Farreids
what I
to the
believe
left, as well as
Mike's
a whip
team
coral. Yeah, it
at
looks like
the
we're getting into
Starport in Washington
maybe a
observed yesterday, where they
higher
saw half
density
EV
zone
movement,
of
which
whip
is
corals.
within
Just as soon as
tolerance,
I talk
and,
about how it's only sponges,
I haven't seen that
we
much
come into
movement,
corals.
so I don't have a
Why are you surprised?
explanation for that, but
Actually, I'm not. You're
that
right.
would account for the swings in Edna that we're seeing.
230 sounds good.
Bridge nav. Another move, please. 30 meters, bearing 230.
Okay, do we have any update from what they're seeing on the ship?
Thank you.
Apparently Brian can hear us, but he cannot really talk to us. That may be difficult. We'll see if I can get some text here.
You know, you get this current pushing you around, and then it's
And
moving around the thing
in
you're
the
trying to
meantime,
zoom
looking
on.
at anything else that I guess earlier there was an
from,
issue with the
Sirius.
encoder as well.
Very dynamic day.
Yeah.
Do we know what that issue was?
All right, let's come out.
And so,
Looks
also,
to be
for those of you
that
who are
all
just joining us,
video streams are back up. But video stream
there's
one
actually a
was
much
not
higher
up
current
earlier.
here than we had expected, or that had
Yeah, so
actually
Brian,
been at the beginning of our
I'm looking
dive.
at
So
internet too.
it's
I
been
don't
really
know if there was
interesting
an
trying to
encoder
keep
per
a nice
se,
zoom, being able to
because of
really
traffic
see
issue, but the
what these close-ups are of the biology on the
stream
surface,
one
because
is
the ROV's been sort of being pushed this way and that.
very good here on
And
internet
you might notice
too.
that
I'm gonna go through the internet.
some of our
Stream
zooms don't quite last as long
I still
as
see
we
interruptions,
normally
but
do.
it's not.
Yeah, I'm
the
pushing
disruptions
ahead
that
at
we
like
saw in the last 2 days.
70% of the power on my
Brian is
thruster is
recording
just
that
moving
audio
forward.
stuff.
Oh, that's not that bad.
and
I think Jeff had
usually it doesn't
Jeff had like
reliable by
100% on to try to like
audio.
stay in place or something.
Is everybody aware
Yeah,
that
if
the audio
we're
is being
head on
fed out
into the
on
current, it takes a little
video
bit less.
Our
Just
conference
not as good as
call?
Jeff.
Oh.
I won't say that. Jeff is very
Here on
quiet.
this one.
But if I take it
How about,
broadside,
getting that
we're
switch there
longer, and it takes a lot more thrust to maintain. You can see the like vortices getting kicked off this sponge stock here. Zoom in
Yeah.
on the stock.
Stand by all.
So we were just hearing from our ROV pilot about
Yeah,
how
it
much
is coming on
thrust
the
it's
video
taking
too
just to
for,
stay in place
The video?
and to move forward.
Can we zoom in
So
on
it
the
really
stock
is
here?
a little bit challenging in terms of current right now.
Get to hear myself about 15
Oh, what is
seconds
that? Is that a
delayed.
tiny little crinoid?
Looks like a very young crinoid, yes.
A little bit of vortex-induced
It's the only thing
vibration.
left.
Got a bit more zoom.
The sponge head is gone.
Take.
Yeah, it's kind of sad. Oh, but you can see the little silica strands that make up the sponge stock.
Yeah, and you know we've collected a couple of these glass sponges during this expedition, and when these samples come up, it is
All right, Brian
the
indicated they're
stalks
still working
are
on that. I'm not quite
it's
sure when
exactly like touching glass.
Bit
Yeah.
closer.
It actually looks a lot like a fiber optic cable.
Yeah, yeah.
That's almost exactly what it looks like.
You wouldn't believe it until you see it.
Yeah.
How someone actually came into the lab and thought we were joking when we said this was part of the sample because
Yep.
they just couldn't believe that had formed naturally and was part of a living animal.
Right, come on up.
It really is amazing. The more I learn about sponges, the more I want to learn.
Deb's going to become a marine biologist soon.
Career change. She's going to come over to the right side.
Yeah. I don't think so. Because look at the geology.
Some more of these manganese-encrusted dead sponges.
Yeah. We are definitely seeing manganese-encrusted dead things. So it's amazing. I mean, really, you can still see this pillow morphology even though it's covered by this manganese crust. It's really quite neat. We've sort of been moving in and out of talus and then back into what look like more intact pillows and then back into talus, and sometimes we see sediment and sometimes we don't. But it's been fairly consistent that at least what we think we're seeing is volcanics covered by manganese crust.
Okay, if I keep it moving, coming at the end of this move.
Sounds good. I think we can start coming more south. I think I'm reaching the...
Okay. Yeah, it's
what is that white
slanting
thing in the
off to
background
right.
on the left?
On the
Is it
left?
Is it just sediment? What is that? You see what I'm saying? Now it's in the center.
0,
It's on the big rock. Maybe it's just sediment.
I don't know.
I think
it might be.
The high point eventually is still more like it's not south.
So we've just been making our way along this
something
track
like,
to the south. We've been slowly climbing a ridge.
210.
Sounds good to me. What does
Ooh,
210
what's on
look
that
like
sponge
to you,
stock, Diva?
Levi?
Oh,
Turn
whoa.
on that. I'm happier kind of staying a little more west of south, but...
Can we have a quick zoom on the sponge stalk, please?
Absolutely. I'm just setting up here.
Are they
Right, come
and
on
there's
in,
stuff
video.
behind,
lots of
like
crinoids or lots of ophiuroids?
I can't tell.
Bridge nav.
Crinoids?
Crinoids,
No.
yeah, yeah.
30 meters.
But they're so
and
pink.
210,
So these are, so there are
0.2
multiple
knots.
crinoids, probably at least 6 living on this sponge stalk together.
I would go with that.
Good copy, thank you.
What's the white thing in the back?
Maybe more. A barnacle, perhaps? Yeah. Yep. So there's a barnacle. I'm not sure what that is.
Is it just some piece of...
There's a little thing with a red end on the right side of the screen.
Any more zoom video?
That's full.
That's it. That's all the zoom we have. I don't know what that
Are you
is.
guys asking about that little orange dot on the right?
Yes,
yeah,
we are.
Is it the end of like an amphipod or
There's
something?
another one.
Yeah, because there are 2 on it.
I'm lined up, 210.
Copy.
No, I think that might be a tip of a parasitic snail.
Oh.
Maybe. There's some parasitic snails that
You
are
limits?
specialized for echinoderms and crinoids, and that looks like them. I see 2 of them.
Make a new one.
Thank you,
There's another
Scott.
one down there.
They have a little, I think, siphon for a mouth that they stuck in and stuck out the fluids of the echinoderm.
A parasitic snail.
They all have them, or most of them.
That was really interesting. I don't know that we've seen parasitic snails yet on this expedition, have we?
No, I don't, but Scott's very correct in saying that these do
Hey, come on
inhabit
up, video.
a lot of different types of echinoderms, these eulimids.
Oh, I...
So it's interesting. We haven't necessarily seen most of the biology that we perhaps expected to see, but we are seeing every so often something new that we hadn't seen yet, like these parasitic snails. Shore-based science team. There's something we must discuss, and that is our science call after the dive. I would recommend that we just have the science call as we usually do, 5 minutes or so after the end of the dive. Does that work for everyone?
Coming in video?
Yes, that works, so long as the telecon cooperates.
That's absolutely
Yeah,
true.
that works for us here at UH.
Great. So we are expected, I believe, to get off bottom in around 15 minutes, although I will ask the navigator for an update on that. And then about 5 minutes after we're off bottom, we will... Oh, look, is that a chrysogorgid?
Very good.
I am so on it today.
So this is likely chrysogorgia, as well as we've got the
squat lobster,
chirostylid squat lobster living inside of the chrysogorgid.
Navigator, this is Watch Lead.
And they're usually the same species as squat lobster.
Are we still coming off the bottom in
living
15
in
minutes?
Living in the
we
same
gotta come off
species
at
of
130 to be on
coral.
deck, be 50 meters at 230.
Perfect. Just wanted to check.
Yeah, we got a watch change at 130 as well. I don't know.
Oh,
perhaps from the
Okay.
genus
We'll probably do the watch change and
Uropticus.
then let them come off the bottom.
Okay, so Shore-Based Science Team, we do have confirmation that we'll be
When you're
coming off
ready,
the bottom at
video.
1:30. Or right around 1:30.
Just a couple seconds.
It's a seasick.
I usually see them waiting
Little
around.
squat lobster there. So it's got to be a seasick little squat lobster there.
Yeah.
All right. Clear.
And for those of you that are, never mind. Oh, I'm still on. Sorry. For those of you that are following along with us today, we know it's been a little bit hard with our video feeds at the beginning of this dive, and we appreciate your patience. And I will also note that we are coming off the bottom a little bit earlier today. We're having a slightly abbreviated dive because on the way down, the ROV actually lost hydraulics, which means that it can't use its manipulator arms and it can't move some of things like the lights. And so what we want to do is come back a little bit early today, which gives our ROV time plenty of time to fix the hydraulics so that we can dive tomorrow. And so hopefully when we do our next dive, which will be at a much shallower depth, about 600 to 400 meters, and when we're there, hopefully we will have better video, and our ROV will be back up to 100%.
Some more euplectellid sponges, hey?
Sorry. Oh, those are so pretty.
I know, I love these.
I think your point about people not really realizing these are animals is a very, very good point. I mean, when you look at them, they just don't look like animals. So
what do they look like?
I mean, it looks like a vase that I would keep at my house.
What is that thing? Is that part of it?
Sure.
A little bubble?
Is that part of it?
So that looks like a little tiny
isopod.
Yeah, it's sort of a bubble.
Bubble?
Huh.
Yeah, it's a bit weird.
A couple little balls of goo or something.
Navigator, is that a technical term? Goo?
Scientific term.
Well, it's refracting our lights. You can see the 4 lights in it.
Yeah.
That's cool.
It's not. I'm pretty sure it's not full of air.
Yeah.
If it is, it's very dense air.
Just a touch more.
When you're ready.
Hi, guys. This is Shirley. I obviously missed a fantastic dive because I'm just getting onto the site right now, and I'm seeing some amazing sponges.
It has been amazing, Shirley.
I'm
you
not
have
gonna
miss the sponge train
This
today.
has been the sponge dive of the expedition.
So for those of you who are just joining us, Shirley Pomponi is another sponge expert. So we've been asking Chris Kelly a lot of questions today, but Shirley is another sponge taxonomist. And she's going to want to replay this dive.
We're coming off the bottom, but we can make another move or not.
What is that one?
Would you rather be
That looks like a
still
Tridopleura.
coming off the bottom? Or
No, because, I mean, there's only one direction I'm going to...
Yep,
Yeah.
correct.
Okay.
Tridopleura.
Okay, I'll do another move then.
Yeah.
So Shirley, did you see that little thing we were zoomed in on before?
Bridge nav.
Right, me
Yeah,
and
one
you
more
on.
move, please. 30 meters, bearing 210, 0.2
Say that again.
knots.
Your audio is really bad on this phone.
I'm sorry to hear that. Did you happen to look? Did you happen to be online when we had just zoomed in
copy, thank
to
you.
that glass
Yeah.
sponge?
I'm going to, I can barely hear you. I'm going to get onto the chat
Come
log.
in.
Okay.
Like there was a really amazing branching sponge that I've never seen before. Maybe Chris has. I'm going to get into the chat log.
Great, thank you. Look, a fish.
Ooh, okay, so this, brilliant. So this looks like a
Catfish.
great, I mean, I'm going to guess Ophidiid, but,
what was that?
A bit more zoom.
or Cuskeel. But it's got some really big eyes. Confusion. I see Bruce Mundy is not in the chat.
Should we kill the lasers?
Yeah, go ahead. Kill
Is that
the
a
lasers.
Chrysogorgia it was next to? I couldn't see it.
I'm not convinced this is an
Lasers
Ophidiad. yet. Confusion.
Are those dangly things part of the fish?
Yes,
Yeah.
they are. And those are likely sensory, play a sensory role.
It wasn't just planning on winning the mustache contest.
Like they have on board? No.
No.
I have no idea what Ophidiad this is. Okay, so Jeff has just jumped in and he says this is Spectrunculus, which is a great name.
Yes, I agree.
Thanks, Jeff. So Jeff Drazen is a fish expert at the University of Hawaii.
Oh.
Nice.
Nice.
Well, that was a beautiful fish. We actually have not seen that many fish today. We've seen a few of those Synaphobranchids.
Yes, exactly.
But really, it hasn't been a very heavy fish dive either. So really, this is the one to replay and watch if you are a sponge enthusiast.
Got a bunch of small stalks here. I don't know if they're coral or what they are. Let's take a zoom here. If you have any more, go for it.
What is that?
So this is another isidid or bamboo coral with
You're
a
off my port
cladorized
side
sponge next to it.
Excellent.
Hands up.
What's that?
You're off to my port
Oh,
side.
okay.
And you can really see that
Wow,
pebbly
video.
manganese crust texture.
Full way.
Another Chrysogorgid there. As well as now the geology looks a lot more like what we were seeing on dive 1. Sort of more rounded
Dive
crust.
1?
Well, not dive 1. Dive
You mean the first dive
first
of the seamount?
geo.
Okay,
Yeah.
I'm like, no, it doesn't look
No,
anything
first
like
geo.
dive
First
1.
geo.
Yeah,
It's changed a lot since we touched down. Yes. Give me a point. Come on. You'd be a terrible teacher. No constructive criticism whatsoever. It's just like, nope, you got it wrong.
I've got nothing to say. Like,
A couple more of these large Poliopogon sponges in view right now, as well as a couple Camachulid crinoids.
They're getting silly.
Looks like we got about 5-6 minutes left.
You want to stow the swing arms? You want to stow the swing arms?
I think they are stowed. Yeah.
Good job.
did that sponge fall over too? Did that sponge fall over too?
This one in front of us?
Yeah. Or is
No,
it
I
just,
think it just looks like that's where it's growing. Look where its base is.
I can't see its base. Oh, and now I see its base.
Yeah.
Yeah, off to the right. Video, this is Watch Lead. So we'll have our science call about 5 minutes after we come off the bottom. So if you could, take the line off the video, that would be great. So in our final remaining minutes of this dive, does anyone want to reflect upon what we've seen and what we've learned today?
She says while staring very poignantly at me. So we've had a really interesting dive today on this flat-topped seamount or geo. We were expecting to see really high densities of quite large sponges as well as quite large corals, because that's what we've seen on the 2 guyot dives we did yesterday as well as the day before. But instead, we've seen something we really didn't expect.
Which is lots of sponges.
Lots of sponges. Not extremely abundant, but by far the most abundant animals we've seen during this dive.
I mean, we certainly saw multiple sponges at pretty much all the time.
Yeah.
So,
and there were about 4 or 5 different types of sponges that were constantly repeated. Pharreids, euplectellids, cladorhizids, some bolosomines, etc. And the geology has also been very different to what we've seen on the past couple dives.
Certainly, it's not exactly the same.
And so it's just been quite surprising, and unfortunately, we're going to have to leave the seafloor soon. But it's given us a little taste of what some of this seamount top is like.
Right. Chrysogorgid?
2 chrysogorgids, or 3 chrysogorgids. And a comatulid. And there's also been quite strong currents, as we've seen. Our pilots have had quite a job maneuvering these currents, as well as many of the animals that live here.
Yeah.
We've seen a lot of dead sponge stalks, likely knocked over by currents. Hardly any corals.
Look at those guys go.
And then here you can see these chrysogorgids absolutely just, waving around in the current. And just not very much other fauna around. A couple of sea stars, a couple of commensals living on the large sponges, but not much else compared to a lot of our other dives in this Marianas region.
And Scott France, who's a coral expert, had asked us to keep our eye out for a specific type of chrysogorgid.
Metallogorgia.
Yes, which we did not find on this dive.
Or on any of our dives so far in the Marianas region. But we still have 4 more to go if you are listening onshore.
4?
3?
3.
3 more. Yeah, we still have 3 more dives to go. And lots more to see during that time.
Absolutely.
Oh, I'm quite sad it's only 3 more.
Me too.
All right, come on in. Color is definitely different on this middle one. All right, come on up.
So do any of our shore-based scientists want to say a final word or two about this area or what we've seen today?
Sponge experts, how have you felt about today?
I'm waiting for Chris to give his overview.
I have a negative.
Sorry, we were having a little sidebar there. Could you say that again, please?
Oh, Chris, we were just looking for final thoughts on our dive.
Everyone go ahead and leave.
So it was surprising the fact that we did see a large number of sponges with virtually no corals. I have seen that before in a few places. I can think of one place in the northwest Hawaiian Islands that we saw this during Hurl submersible dives, but it's been a while. So it's curious for sure. So that's one thought. The current is strong, and we were expecting a bit of a current here anyway. That's why we chose this ridge, but it was stronger than we expected. I guess that's it, and I don't know what else to say. Very spongy place.
Right, one thing I will add is that, we're not all that far from the last couple of seamounts, I don't think, that we've been looking at. So it is kind of fascinating to see such...
So let's keep your heading right now.
And as Chris, Diva, Deb, you've all said, this lack of corals, given the topographies that we saw on the maps when we were deciding where to dive is kind of a surprise, and given what we've seen in the last couple of days, it's a surprise. So exploration keeps surprising us.
Yep, and that's a really great point to end on. And we may not know what we're going to find, and that's the reason we come out here.
The bottom.
All right, so it looks like we're leaving the bottom. For those of you that have been following along today, we hope you've enjoyed it. And tomorrow we will be doing a much shallower dive, looking for commercially important fish species and
coral species.
Precious coral species, which are the kind that are made into jewelry. So please tune back in, and we will see you tomorrow.
Shore-based scientists, we
We'll
will
see
speak
you
to you
in
in 5 minutes.